Thank you, Dennis. Happy to talk today about some introduction to understanding how we do meta-analysis in genome-wide association studies, or GWAS. A lot of what I'll talk about today is more generally applicable to meta-analysis in general, and is just what we apply in the specific case of combining GWAS. The hope is to provide both some intuition and a little bit of math about how those things work, and a small example in terms of how this actually gets implemented in software for people looking to do this kind of meta-analysis of GWAS results. So our basic interest in meta-analysis is if we imagine that you've taken the time to collect some data, do a GWAS on your trait of interest, and then you find out that there's other data that is also done at GWAS on your trait of interest. So you now have two different result sets. Potentially, there are many other people who have also done GWAS, so maybe more than two. But in the simple case, you've got these two different GWASs, and you want to find some way to put together those results to get one more single cohesive picture as far as your best possible GWAS for looking for these common variant effects on your trait. And so this really is our core motivation, is how can we combine the results from these different studies in order to have a combined analysis that's more powerful in the statistical sense, that gives us more precise effect sizes, and really does our best to pool that information to give us the most complete, up-to-date version of that GWAS. And the kind of core idea for our approach will be that if we have summary statistics from each of these studies, then we can combine them in some way that weights by kind of the strength of evidence from each of the studies. So you can imagine that if I did my GWAS in 1,000 people and you did your GWAS in 10,000 people, maybe you wanted to give more emphasis to the set of results from your study on the basis that we might expect it to be more informative. And so kind of one key thing to note here is that we're focusing on the case of meta-analyzing based on summary statistics rather than pooling individual level data to do some kind of combined analysis. And so there's a downside that we lose a little bit of power that we might have versus if we pooled the individual level data and did some kind of single analysis of that full set of individuals. But we gain some protection in terms of study-specific artifacts that we would otherwise have to control for, in terms of making sure we had really clean data that was directly comparable to do that kind of single joint analysis. So by doing this kind of combination of summary statistics at the meta-analysis level, we're allowing kind of study-specific factors to be kind of controlled with that either case control or association comparison within the study. So like study-specific shifts in, say, the mean of that distribution of the phenotype are less important to worrying about our results in the meta-analysis. All right. So kind of from this core idea that we want to kind of pool our results from the different studies, our statistical approach will be more or less a simple weighted sum. So the math's here if you want it, but so the idea is that we have these test statistics, in this case z-scores from each study, those z sub i's, that we're then going to sum over them to create some weighted sum here based on these weights w. And you normalize out those weights and you get some overall test statistic. So here in terms of kind of an overall z-score. And so the simplest case is the one shown here, where those weights are proportional to the square root of sample size. So again, that idea that the higher sample size study might deserve more weight. If we assume kind of all else equal about the studies, that there should be more information there. Kind of the other most common approach, at least in GWAS, is that we instead weight by the 1 over the standard error of the beta effect size estimate from each of these GWAS. So that beta being either your kind of direct effect size at a linear regression for continuous phenotype, or your log odds ratio from having done logistic regression for a binary phenotype case control analysis. But so kind of weighting directly by our estimate of how much noise there is in that estimate, and we do 1 over, so essentially more noise means less weight towards that. And so kind of that weighting by the standard error versus weighting by the square root of the sample size, we expect to be more or less equivalent. Again, kind of this world that all things are equal between our different studies. They should both give optimal results and converge to the same thing under this assumption that there's kind of the same true effect size and same study design for each of these studies. In practice, we often just stick with the inverse variance version, since that gives a little better capture of differences in power between the studies that might scale more with, say, allele frequency or case control balance within the study, rather than directly with sample size. But so kind of at the core of meta-analysis for GWAS and for meta-analysis in general is this kind of weighted summing of the effects to get an overall estimate of the effect. So that's the kind of underlying math. So now let's talk a little bit about how we might actually accomplish this in practice. So there's a few different common packages for performing meta-analysis on GWAS results. The one I'll talk about most today is this one called METAL from the University of Michigan. So Custis' group is nicely documented, has a solid history of use kind of in many of these large GWAS meta-analyses, and provides some nice interface. Other options in the space include GWAMA and Metasoft, as well as always the option to just roll your own calculation for the same kind of basic idea of performing a meta-analysis. We'll talk a little bit more at the end about kind of extensions in different models and some places where there might be some variability in using these other pieces of software. But for METAL, kind of the two key things you need to be able to perform this meta-analysis is you, of course, need your GWAS results from each of the different studies that you're going to include in doing this meta-analysis. And then you need a script specifically for METAL that kind of defines the formatting of those files and how you're going to put them together into this meta-analysis. And I think it's really important to focus for a second on just the kind of work in advance that happens to preparing that GWAS data to be ready to go into the meta-analysis, since that tends to be where a lot of the work is in these studies, is in getting all the GWASs prepared for meta-analysis rather than the actual computing the meta-analysis results themselves. And so we can imagine the kind of the first most simple step is making sure you've identified the relevant set of studies for your phenotype of interest, and have those ready to go, and certainly have got the data shared from those different cohorts. But also this kind of key feature of making sure that you've made sure that you've got clean, uniform data to work on. So this is in terms of making sure you've got phenotypes that are defined equivalently. So we can imagine if we're doing a GWAS of height, and we're going to do it on the effect sizes weighted by their standard errors, then we want to make sure that all those effect sizes are in comparable units. You don't have one study reporting, say, an effect in terms of inches and another reporting effect size in terms of centimeters, because then the scaling is wildly off, both in terms of the effect size and in terms of how much relative weight they're getting based on their standard errors. We also want to make sure that you have the variants from each of those GWASs identified in some consistent way, and that they're kind of consistent in terms of human genome build. So when you go to line them up in the meta-analysis, you can take, in METAL's case, matching on their RSIDs and being comfortable in knowing that you're then capturing results for the same variant across all the different studies, and that they're all consistently lined up in that way, making sure they have concordant alleles, particularly in the case of making sure that you've managed the names on any multi-allelic markers and things along those lines. And also that they've come from a GWAS with a consistent analysis method. So kind of similar to, say, inches versus centimeters issue. In the case of a continuous phenotype, for a binary phenotype, you need to make sure that you have comparable effect sizes. You don't say have a mix of people who've done a linear mixed model versus people who've done logistic regression, where again, you get kind of a scaling difference on those effect sizes. So kind of all this effort that goes in up front to make sure you have nice clean files that are consistent with each other that you can then plug into doing this calculation across the different results for each variant. I guess so the last piece that's listed on here that kind of fits in there is also any additional QC filtering you want to apply beyond what filtering may have been done to raw summary statistics from each GWAS that are shared with you. So for instance, you want to impose stricter filtering on minor allele frequency or amputation quality or things along those lines on a per study basis. In terms of what you allow into the meta-analysis, then you want to make sure that you are also know what you're going to do on that front. So once you've done the work to kind of put together your set of files, then you can put together the script for metal. And so the general layout of this is you start with this header block that defines some general overarching information in terms of, you know, if you're going to have white space or tab delimited files or CSVs or whatever is your input, and how strictly to treat the columns of those files. The option to apply genomic control as part of your meta-analysis, generally that's something that's recommended against in more current practice, but historically has been of more interest in terms of ways to control potential inflation in the GWAS, genomic control being perhaps a little more strict than it turns out we want, for at least for polygenic traits. And then setting up kind of this overall tracking of allele frequency information, if that's something you have from each of your studies, will give you a good kind of post-hoc check that you've done a good job of lining up the variants in terms of tracking both the average allele frequency and the minimum and maximum of that to verify that you don't have likely allele swaps or strand flips that are essentially reversing the direction of the allele one of your cohorts. So if one cohort shows up in the meta-analysis as having MOF of 5% for a variant, or sorry, allele frequency of 5% for a particular variant, and then the other study shows an allele frequency of 95% for that same allele. In the other study, you probably had some sort of flip of the alleles. And if you don't correct that, then it's going to look like you've reversed your effect between the two cohorts, and thus will wash out any signal. And the last line here is to define how you want to do the weights in that kind of weighted sum sort of meta-analysis. So the scheme here is the kind of inverse variance weighting, where you're using standard errors, and the alternative to do sample size weighting is just the alternative sample size scheme, and specifying that you're going to use that sample size column that needs to exist in each of your input files. So with the initial setup, then you do a series of blocks describing the format of each of your files, where the key here is to identify what the column headers are for the fields that are needed for the meta-analysis. So identifying which column has your variant identifier, so RSID or whatever you're using there in terms of the marker, the two alleles, where the first allele should correspond to the direction of the effect, your allele frequency field, your actual effect size there, with the option to take log of a column that has odds ratios, as shown here in this example, the standard error field that you're using for weighting, and then this line to say to go ahead and process that input file based on that description of what the header is. And you do this for each of your studies. And once you've done that for each of your studies, at the end you specify what you want your output file to be, and tell it to go ahead and analyze, and go ahead and add this heterogeneity flag, which will then include, as part of the meta-analysis, the set of diagnostics for potential heterogeneity between the different cohorts. And so once you've got that script together along with your files, it's a single command line call to the metal program that is just, the metal program, this input script you've written, and where you want to produce a log, run from the command prompt. For your genome-wide data, running a single machine, you're talking, order of a handful of hours, maybe, if it's really big, in terms of genome-wide data. But what you'll end up with is this log file that will contain lots of both information in terms of if you've set up any filtering, how many things were filtered out, any kind of issues with discordant alleles between the different input files, or other kind of invalid values that might have popped up and caused errors. Then your kind of core output file, this text file of the actual results of the meta-analysis, which will look just like a GWAS results file in terms of having the variant and its estimated effect size and p-value, and also these heterogeneity diagnostics, all those kind of things. And then this kind of summary info file that most informatively is there to give you what direction the different input GWASs were read that will correspond to a field in the meta-analysis results that nicely gives you a string indicating what direction the effect of a given variant was in each cohort. So it's kind of a visual summary of how consistent the direction of effect is. So kind of at the most mechanical level, that's at least one example in terms of using metal of how we can go ahead and run this kind of meta-analysis. And so kind of the last piece there is once you actually run your meta-analysis is to circle back and verify that things actually look reasonable and consistent in those results, not just jump to the p-values and either jump up and down excited over your significant results, or give a sad face to your screen when you see no significant results. So the first key is to check do these kind of consistency checks. So one is that check I already described in terms of allele frequency. So if you see these variants that have kind of wide variability and maximum and minimum reported allele frequency, or the far ends between 0 and 1, that maybe there was some issue in terms of how allele frequency was, how the alleles were identified, and lining up those alleles for meta-analysis, making sure the effects were all lined up to correspond to the same allele. The other thing that can trip this up where you're perfectly fine on aligning the alleles, but one of your studies has reported just a flat minor allele frequency rather than the frequency of a specific allele, and thus it's getting flipped inappropriately in that tracking of the allele frequency. The other note here to be aware of the population is in the sense that if you're performing a meta-analysis with say multiple populations or more diverse sets of populations, which arguably you wouldn't want to do in this kind of basic fixed effects model that Metal is running. But if you are doing that, you may see more variability in that allele frequency as reflects kind of differences in the population balance rather than any issues with one results. So need to, of course, as always, take the specifics of your data into consideration when evaluating these kind of QC and consistency checks. The other piece here, though, is for checking for heterogeneity of the actual results between the different studies. So there's kind of two primary things here. One is the plain test statistic for heterogeneity, which Meta reports as Cochran's Q, along with an I-square, which is roughly speaking an effect size for the heterogeneity. And so what this Q is a test of whether there's signs that the true effect size is not the same across all of the input GWAS, essentially on the basis that the effect sizes in those input GWAS are more variable than they should be relative to their estimated standard errors or sample sizes. Empirically, this tends to be an underpowered test. So its power is more or less proportional to the number of studies included in your GWAS, in your meta-analysis. So if you're doing a meta-analysis of, say, 5 cohorts, it's only really got the comparison between those 5 numbers to identify whether there's excess variability, and that's unless there's kind of extreme outliers, you're unlikely to catch significant heterogeneity there. But it's something that's still worth a check, and there is that I-square kind of effect size measure of the amount of excess variability. The other piece is to look at this more visually, and this in particular is useful once you have some loci that are significant in the meta-analysis to go back and verify that significance isn't being driven by, say, one particular cohort or a couple of particular cohorts using a forest plot. And so that looks something like this, where you're taking essentially the effect size from each of your individual cohorts here. There's something like 20 cohorts on the slide. And lining up the effect size estimates from each of those cohorts, along with their standard errors, to see if they're mostly consistent or if you've got specific outliers that might be dragging that meta-analysis result around. So this is one particular case where there was an apparent outlier affecting that overall effect in a way that's not modeled by the meta-analysis, and assuming they kind of all studies are created equal and this just summing across them. So a good follow-up visual check that perhaps gives you a little more clarity, especially on individual outliers, than you might get from just the overall Q statistic for if there's overall heterogeneity across the cohorts. So that mostly gets through kind of the stock standards of kind of basic inverse variance weighted meta-analysis. So I wanted to conclude here by taking a little bit of time to run down kind of all the different ways we might go beyond that kind of really general basic meta-analysis model for GWAS. And kind of the top one here is a random. The fixed effects assumption doesn't apply, that there may not be an identical true effect size across your different studies, whether that's due to their various study designs or populations or whatever's going on there. Then the kind of single step generalization is to include essentially a random effects term that says that the true effect varies between different cohorts, and then you're trying to model as part of your meta-analysis what the mean of that distribution of true effects is. And that's what gets labeled as kind of this random effects meta-analysis. So there's both the kind of classic random effects meta-analysis and then some extensions that try and generalize in terms of doing a combined test that essentially says a SNP deserves to have some sort of association to phenotype, either if the kind of mean effect is non-zero or if there is some variability in it, and thus it's non-zero at least somewhere, and kind of combine those hypotheses. And that's what this RE2 model from Han and Eskin that's included in Metasoft does. Metasoft also includes kind of the more standard random effects meta-analysis and a few other different variants of meta-analysis. So if you're looking for some of these more complex or more generalized models, that's one of the places where Metasoft starts to become preferable to METAL as kind of the older standard. Another place where we might want to kind of expand upon the basic fixed effects model is if we're specifically doing meta-analysis across GWAS from different ancestries, and some tried transethnic modeling. And so it's possible to do this and just treat it as a random effects model, but another alternative is to try and more specifically model the population differences. So if you know in advance that you say you've got 10 cohorts that are of European ancestry and 10 cohorts that are of East Asian ancestry, and you have some kind of population genetics knowledge of how different those two populations are, then you might be able to plug that into a more sophisticated model for taking into account the kind of within versus between ancestry variation. And so I think one of the main names here is a software called Mantra from Andrew Morris. That's a Bayesian model for doing this kind of thing. And there's also more recent follow-ups from the Morris lab as well, and kind of frequentist framework as well, getting at this idea of trying to more specifically model the special case of transethnic variability within the meta-analysis, rather than just treating it as a straight random effects model. So the next point here is in terms of effective sample size. So this is in cases where so we talked a lot about the importance of making sure you have equivalent scaling of your effect sizes. So in some cases, you've got studies from, say, enough different study designs or enough different analysis models that you just don't have a way to get consistent scaling of those effect sizes. And perhaps with the difference in analysis models, it's not quite right to even just take their pure sample size as kind of the index of how much information is there. Because, for example, if you have family-based data, then depending on the degree of relatedness within the families, you get different statistical power. And so this is a case where you may end up wanting to do some sort of approximation of an effective sample size that tries to get more consistency in terms of a sample size-like quantity that gives you a more constant index of the amount of information in those different studies to define their weights. And there's less specific software for this. This generally requires a little more heuristics and knowing the specifics of your analysis model to adjust based on, to figure out what the appropriate calculation is for their effective sample size. But this at least is one way to handle those kind of situations where you have maybe some more unusual data sets in terms of how the analysis has been done or how the study was designed. One of the nice advantages in thinking about something like an effective sample size-based meta-analysis is that kind of this core idea of meta-analysis as a weighted sum of the results from your different studies. If you're operating on, say, the sample size weighted sum of the z-scores, is that as long as the individual studies themselves are properly calibrated for their z-scores, then improperly calibrated weighting, so if you're kind of off in your effective sample size weight, will only hurt power. You're not going to induce false positives. So, given that the null is properly calibrated for each of those individual z-scores, how you sum them together, as long as you're not directly weighting them by how strong their z-score signal is, any kind of weighting that's blind to that is going to still give you a properly calibrated null and maintain your type 1 error rate. Then just becomes a question of how efficiently you're giving weights to the studies with more statistical power. And the last piece here is we can think about meta-analysis not only for a single trait, but we might want to start pooling related traits. This is an area that's seen much more active development in terms of methodological and statistical work in the past few years. So there's just a few such examples on the slide here: a CPU soak and a method from Blorma et al. And ACID and this extended random effects model, and certainly plenty of other players in the space where a lot of the differences come down to more just thinking about once you're thinking about meta-analysis across different traits, how you want to be kind of formulating your hypothesis there, whether you're looking for trying to test whether variants have an effect on all of those traits, or if you're looking for variants that have an effect on some subset of those traits, or whether you're trying to find things that are at least variable across those traits in kind of the random effect sense, and, regardless of the mean effect of the SNP, that there are signs of pleiotropy and non-zero effects across some of those traits. So, in that space of ways we might think about what the hypothesis is once we're talking about multiple traits, there are many different options in terms of thinking about how we might perform a meta-analysis to pull the results from different GWAS studies to get a test of that hypothesis. So hopefully that gives you at least some introduction to how we think about both the kind of statistical side of meta-analysis and how we can actually implement it in practice. So again, it's kind of this core idea in the GWAS sense and also in the general sense of meta-analysis that we have results from multiple cohorts or multiple studies, and we want to combine them, and we can do that with this kind of weighted sum of their evidence, with the key decision point being how to define those weights. And just to reiterate kind of the importance of making sure that we have kind of carefully curated the input files going into Metal or other choice of software to actually perform this analysis, and that we're actually being careful to verify kind of the consistency of those results and how they hold up in terms of looking for heterogeneity and things afterwards in terms of these kind of sanity check on analysis results before we embrace that final p-value. And then there are many directions that maybe we may think about if we want to get away from that fixed effects model and being more nuanced in how we combine studies from the many different sources now that are available in terms of the summary statistics from these genome-wide scans, and making sure we're putting them together in the most informative way based on the specifics of the different studies. So hopefully that's been helpful, and appreciate your time in thinking about GWAS meta-analysis.